Hi, and thanks to RMS for hosting this and having me. My name is James Major. I'm from Northern Arizona University. I'm currently wrapping up my master's and will graduate in July. My talk today is about utilizing existing conservation policy to fill river protection gaps. The nation's wild and scenic river system in the U.S. was established to protect free-flowing rivers and their outstandingly remarkable values, or ORVs. When a river is designated wild and scenic, up to a quarter-mile buffer zone is also protected to ensure the maintenance of these ORVs that are river dependent. Today, in the U.S. and Puerto Rico, there are 226 wild and scenic river designations, totaling over 13,000 miles. Clearly, there's a lot of room for increasing the number of protected rivers in this system. Fortunately, there's a catalog of rivers that have already been identified as potentially eligible for designation into this system. The nationwide rivers inventory contains over 60,000 miles to choose from. The question is, with so many eligible rivers, how do we know which ones would be the best to protect, considering that we have limited fiscal and human resources? To that end, this study asks, where are NRI reaches concurrent with gaps in protected areas that are managed for biodiversity? I used geospatial software and R language scripts to manipulate interagency data from the Forest Service, USGS, Park Service, and EPA to answer this question. It is generally understood that in the Southwest, 80% of all animal species reside within riparian corridors, but less than 2% of the land is riparian. Because the desert Southwest is the most vulnerable to climate-induced water scarcity, this study focuses on the EPA Level 3 Arizona-New Mexico Mountains ecoregion. This ecoregion, as its name suggests, includes parts of the states of Arizona, New Mexico, and also Texas. The NHD contains spatial information and flow characteristics about each surface water feature in the United States. Wild and scenic and NRI datasets contain information about ORV designation and eligibility status. To make spatial comparisons within the ecoregion, I, along with a lab mate, combined the wild and scenic, NRI, and national hydrography datasets. Once we had the data combined, I could then address the research question by performing a gap analysis. A gap analysis is accomplished by overlaying three datasets: location of protected areas, species range, and land use data. In addition to these datasets, I also added the hydrological dataset so that gaps in protection could be compared with the locations of NRI reaches. The USGS maintains a database that catalogs protected terrestrial and marine areas. Areas with the highest level of protection that manage for biodiversity while allowing for natural disturbance to occur, such as wilderness areas, are ranked as GAP Status 1. Status 2 protected areas are also managed for biodiversity, but ecological disturbance may be suppressed. The other two statuses are not managed for biodiversity. Due to the aims of this study to identify river reaches for biodiversity, I limited the analysis to areas containing GAP statuses 1 or 2. Because the species range data for aquatic organisms is still in development, proxy species, such as aquatic-dependent reptiles and amphibians, can be used instead of full-time in-stream inhabitants to assess gaps in riverine protections. For this gap analysis, the narrow-headed garter snake serves as that proxy. The narrow-headed garter snake has suffered large population declines within the past 20 years, leading to its listing as endangered in 2014. These snakes are unique in that they are not usually found more than 200 meters from the water's edge and prefer flowing water where they can access the soft-finned fish that make up their diet. These habitat requirements make them particularly susceptible to alterations such as the development of reservoirs and degradation of riparian species. These characteristics make the snake an excellent proxy for the riparian obligates in the ecoregion that rely on free-flowing rivers. All lands unlikely to support snake populations were removed from further analysis. Next, I isolated perennial streams by selecting only the rivers within the reach that had a perennial reach code. The map on the lower left shows the snake's range in tan, according to the GAP project. Since these snakes rarely venture more than 200 meters from the water's edge and rely on flowing water, much of the identified gap range is inaccurate. To address this, I created a quarter-mile buffer layer approximately double the expected range of 200 meters. This represents a truer approximation of the snake's range and excludes areas that are not expected to support a snake population, such as non-perennial reaches and uplands outside the observed hibernation range. I found that the majority of the perennial rivers in the ecoregion are within the narrow-headed garter snake's range, over 78% of them in fact. Looking at the map, we can see the ecoregion is in the heart of the snake's territory, which makes the persistence of perennial rivers so important for this species. Further, I found that nearly 3,000 miles in the snake's range is perennial. However, only a little over 600 miles, or 22% of perennial river reaches, are protected as either gap status 1 or 2. These are the adequately protected reaches for the narrow-headed garter snake. Taking into consideration that only 22% of suitable habitat falls within areas protected for biodiversity, that leaves the remaining 78% of suitable habitat as the conservation gap. This gap is quite large, especially considering the endangered status of the narrow-headed garter snake. In case you were wondering, I found a negligible percentage of the rivers within the snake's range that have an incompatible habitat on both banks of the river for an entire NHD reach. Think about the NRI as being a repository of rivers that can be tapped for further conservation efforts. 580 miles, or 26% of the rivers seen in this map, are NRI rivers in the conservation gap. These rivers are not included in the PADIS as having any sort of conservation status for biodiversity protections. To reiterate, 22% of rivers that are suitable snake habitat are adequately protected, while 78% are not. Within the conservation gap, 26% of the rivers are on the NRI. With such a large proportion of the snake's range in the ecoregion located in the conservation gaps, it seems necessary to increase protections. Designating NRI rivers as wild and scenic could narrow the gap for the narrow-headed garter snake and for the aquatic species for which it serves as the proxy. In fact, if all NRI rivers were designated, the gap in protection would be reduced by a quarter of what it is today. The results for this study only encompass a single ecoregion. There are over 100 EPA Level 3 ecoregions, and all could benefit from studies like this one. Conducting these analyses on an ecoregion scale can ensure that unique ecosystem services within that ecoregion are identified and targeted for conservation. Thank you for listening today. I want to thank the Arizona Water Association for a financial grant that allowed me to get the equipment that I needed to work from home at this time, and also RMS for hosting this and allowing me to get in with all of you. And now we will open up for questions.